For the introduction. Indeed, I work here, so I don't need to introduce the Eindhoven University of Technology. So I'm from the Signal Processing Systems group of the Electrical Engineering Department, and I lead the Biomedical Diagnostics Lab. We work on signal processing for many applications, and well, talking about and discussing about sensors, I thought that perhaps electromyography could be an interesting one with several different applications, again, also within this field. I will not really, we are not working on new sensors themselves, but really on new applications for sensors. So that means new measurements using the sensors that are available or developing technology for emerging new sensing solutions. I realized I put together really a lot of slides, so I will try to quickly jump into the topic. So we are talking about electromyography. That means the activity of muscles. So this is what we want to measure, and first of all we should make a distinction between two types of muscles. We have the striated muscles and the smooth muscles. So these two types of categories have some major distinctions, and if we want to monitor them, of course we have to know a bit about the physiology. Here I'm first of all striated muscles, which are mainly our skeletal muscles that relate to our movement and posture, are, let's say, controlled and guided by innervation. So there is a neuromuscular junction here that will let the action potential propagate through the muscle and therefore elicit the contraction of muscles. So innervations. Instead, smooth muscles, and here I'm giving an example from the uterus. We will in fact dig a bit more into this type of applications for pregnancy monitoring. In smooth muscles, we don't have really innervations, but we do have pacemaker cells, so they kind of auto-control themselves, regulated especially by hormones. There is, of course, skeletal muscles are mainly in their anatomy develops in fibers. That means that there is a dominant direction of propagation of this electrical activity through the muscle. So there is and therefore the action potential will propagate along this direction. This does not happen in smooth muscle, where the propagation between cells occurs through the so-called gap junctions, which can activate or deactivate also. For instance, if you're talking about the uterus, depending on the phases in the menstrual cycle or during pregnancy, they will especially create junctions at the moment that contractions need to propagate. So this is also skeletal muscles are made of bundles of fibers. Instead, smooth muscles, especially the myometrium, so the muscle of the uterus, is a complex multi-layer structure where signals through these gap junctions propagate in really in 3 dimensions, and it's not easy to predict in which direction they're actually propagating. Also, the signals that we measure have a totally different bandwidth. If you are talking about skeletal muscles, then we will measure on the surface a bandwidth of 20 to 400, 450 hertz. Instead, the electrohysterogram, which is the electrical activity of a smooth muscle like the uterus, has a very low frequency, as you can see here. And this is very challenging also on the electronic standpoint, because you have a lot of noise at these low frequencies and a bias and wonder of the bias that makes these measurements quite complicated, also on the electronic standpoint. Why also these very low frequencies is because the action potential that propagates, which makes the frequency at the position where we measure it, is very slow in this case. So we are talking about propagation of centimeters a second versus meters a second in case of skeletal muscles. So that also is reflected in the frequency that we measure. Now, what is an action potential? What is what we measure? In fact, this is created by channels that open across the membrane. These are sodium channels and potassium channels, and they will create a change in the potential across the membrane for instance, of the sarcomer in the case of skeletal muscle, of the muscle cell in general, also in case of smooth muscles. So this is actually something that occurs in both cases. We have these action potentials regulating the activity and contraction of muscles that apply to both smooth and skeletal muscles. And this is what we measure, in fact, from the surface, this propagating action potential. And these are create the onset of contraction, the mechanical contraction. How do they do that? Well, as this is also comparable for smooth and skeletal muscles, what happens is that as the action potential comes, and what happens, for instance, in a skeletal muscle, but also in a smooth muscle, is that the concentration of calcium will increase inside and will enable the binding between actin and myosin that you see here, these filaments, so the thin and thick filaments. If they can bind, because the binding is set free thanks to calcium connecting to troponin, that means that troponin was actually blocking, for instance, skeletal muscle, the actin. Therefore, it could not bind to myosin. But if this binding are set free, then they will start contracting and shortening the contracting element, which is this one, is the sarcomer. So everything is controlled by calcium in both cases. We can find some distinction in the sense that skeletal muscle are mainly actin regulated and smooth muscle more myosin regulated, also because the way calcium increases the concentration in the cell is slightly different. As I already told you, also the geometry and the architecture is different. So the sarcomer and what you see in skeletal muscle will create a shortening in one dominant direction, because everything follows a very regular bundle architecture in one direction. This is not the case in smooth muscles. So what you will see is that these small cells, which are rather smaller, will change their shape and during contraction. That means that also the geometry that they can create is different between contraction and relaxation. So where are smooth muscles present? We will concentrate on the uterus, so the myometrium, which is the muscle of the uterus, but they are also present in the sphincters of blood vessels, stomach, intestine, bladder, and of course uterus. So quite a large set of muscles, and that is why also understanding about the activity of smooth muscles can be quite interesting. I think it opens up an entire field of applications and ways of generating data that are very useful to assess the physiological condition. So this is the outline of my presentation. Will be first, I'll be concentrating on electrostereography, so the activity of the uterus, so a smooth muscle. Then something about electromyography, and we will mainly focus on ways to elicit and measure neuromuscular reflex. And then some first experience that we are doing now with contactless electrodes, sensors that are capacitive. And in this case, the application will be more in the direction of electrocardiography. Well, it's still a muscle, if you think, but the signals are larger, and it's a bit easier to tackle as a first application for these sensors. So electrostereography, why? Well, it's mainly addressing the problem of preterm birth. This is a quite serious problem with a very high incidence, and in particular accounts for 75% of perinatal mortality. And if you think, this type of problems also relate with very severe morbidity, because if something occurs in these very early moments, then the consequences can, in fact, last for the entire life. That's why this is a very critical time, and that's why it's very important to be able to also predict the risk of preterm birth, and therefore be prepared to deal with that. So now, why are we focusing? So what is actually happening in terms of uterine activity? Well, we have, in fact, the source of everything will be the electrical activity that stimulates the uterine contraction, because the action potential will create this increase of calcium concentration that makes the smooth muscle contract and create the contraction of the myometrium that is actually pushing the fetus outside the chamber here, outside the uterus. So there will be an increase of pressure as a result of this contraction. The cervix will dilate, and the fetus will be expelled at the very end through the birth channel. How is this process monitored nowadays? Well, there are two possibilities. There can be used an internal pressure catheter. This only, of course, when the water is broken and the membrane are broken, and you can insert the catheter inside the cavity over here. Or you can use the tocodynamometry. That means, actually, from the surface, you sense contraction of the belly by just using a strain gauge, so that will sense stretching of the skin here on the surface. You can imagine this is rather inaccurate because any abdominal contraction will also contribute to that. But still can be used, for instance, to look at the frequency of the contraction. Amplitude is quite difficult. So in this area, what we introduced is the use of electrohysterography. So really the analysis of the electrical activity as a first sign in order to characterize the activity of the myometrium and uterine contractions. So compared to the method that we have seen, we hope to achieve accuracy that is superior to the tocodynamometer, and in particular is non-invasive, so it's quite comfortable to apply. So what we will do is to place, so this is an example, we distribute electrodes on the abdomen. There are now a bit more fancier, let's say, solutions with a single patch that contains a few electrodes by which the electrical activity of the myometrium can be measured. In order to characterize this activity, well, basically these are the contractions, so the times that happens every 5-10 minutes. So the timing of the uterine contraction, but underneath this contraction there are actually electrical bursts. So this is the activity that we measure with these sensors. It's not just bias changing, but it's really these are coming really in bursts of electrical activity. It's important to know the amplitude, but also the duration of the contractions, and of course, as I told you, the frequency. So the first problem that we addressed was, where are we able, by just looking at electrical activity on the surface, to predict and assess the intrauterine pressure so that we can replace the catheter, the use of internal catheter. So in fact, we have proven by comparing, this was in 10 subjects, the estimators from the electrical activity that we used, which are mainly based on the first normalized moment of the spectral distribution of the signal that we measure. By this parameter, we could actually predict quite nicely the intrauterine pressure. So this is in fact evidence that indeed the use of electrical potential is a quite promising approach in order to measure intrauterine pressure and therefore uterine contractions in general. And this was, of course, during labor. In order to improve our modeling of the full of the signal, well, we really looked at the full chain. This is a multi-scale model that we developed within an Erasmus Bio Plus European consortium, starting really from the cells and then the ionic concentration at this level to tissue up to the organ scale, and where eventually you see all the layers that we have from the myometrium to the skin, and here we measure the electrical activity, and we want you also to look at the volume conductor in between in order to interpret the signals. So this is indeed this is the full model where we have here the myometrium, so the tissue level with all the cells and then all the layers up to the electrodes that we had on the surface, where we developed our model based on Poisson equation, just and with a number of conditions for continuity of the potential, and this was our source, again based on this cells with a certain velocity, conduction velocity of the action potential through this very simple chain of cells. You see that the size of each cell is a bit smaller than 1 millimeter. And we were able to create signals that indeed, generate signals that indeed are quite alike what you would measure by electrostereography. So we want to really to develop a simulation of the full chain from the cellular source up to the electrostereographic signal that we measure. Now, how can we use all this knowledge in pregnancy? Now we have seen that in labor we can estimate the intrauterine pressure, but that was not really our main goal. Our main goal was actually to be able to predict the risk of preterm birth, and this is very complex indeed. There are several ways over the years that this problem has been approached. You can use single channels, so just one electrode, or multi-channel. And then if you look at single channel, there were very first approach you see in the 80s, looking just at spectral features. Then very recently, in fact, we introduced the possibility of looking at entropy measures to look at the regularity of the signals. But also multi-channel, then you look at the similarity or connectivity between the signals at different channels. So there was no linear correlation already introduced a few years ago, and even earlier, the way we started approaching this problem was to assume a linear propagation of the action potential through the uterus, like really plane waves that are propagating. We thought probably, let's say, up-down, in order to help pushing the fetus outside, approaching labor. So focusing on the first one, these are the type of measurement we did. So this is an electrode that contains 64 channels, so it's at high density. As you can see, only 4 millimeters distance between electrodes that have a diameter 1 millimeter. We band pass at very low frequency, as you can see here, just above respiration to avoid this type of artifacts, and below 1 hertz. And of course, we could downsample because we are at very low frequency. And this is actually the very first wave that was measured and that we could also show. For the first time, you could really see the uterine contraction in terms of electrical activity that propagates through the uterus underneath this electrode. So you see, the red part is is higher energy, and you see how it's propagating clearly in one direction through this matrix. So based on this, we could develop some models to look at the delays between the channels and therefore estimate even direction and velocity of these waves by using a maximum likelihood approach. This was a rather simple model that we used, and these are the results. Well, what we could predict was imminent labor. So if the lady was in labor, looking at the velocity, we could appreciate an increase of velocity and even a dominant propagation direction top-down. However, this with a different sensor that is now commercialized by Nemo Healthcare, this approach did not lead to prediction of preterm birth and not even labor within 1 week. So why is that? So this was a bit disappointing because it was a big trial that we did with 120 patients included. It was one of the reasons could be, of course, that everything I had shown until now was in labor with very strong signals, very strong contraction, very good signal-to-noise ratio. These contractions are measured even at 30 weeks, so very, very early. These are all patients that approach the hospital because of preterm contractions. So, okay. And that is can be one reason. And the other one is that our linear propagation was appreciated in a small matrix covering only 4 centimeters. Here you see these electrodes are large, and most probably we don't observe a linear propagation through these larger electrodes. So this can be also another reason for this approach. So the failure of this approach to predict preterm labor. So what we did later on is to actually focus, as I told you, on entropy measure, sample and approximate entropy. Look at the similar and also extend, this was in single channels, but we also extended that to multiple channels. So look at the cross entropy between different channels. So it's basically looking at the regularity of the series between different channels, and you expect that if there is a contraction, the regularity, so the entropy decreases because everything is well regulated. Otherwise, you see a higher degree of entropy in the signals. So this was between couples of signals, or here even comparing all the channels that we had available, so a multi-channel approximate entropy and sample entropy. Basically, it's a similar approach as the single channel, only now we have a multi-dimensional signal that we input. What we introduce also is when you measure entropy, you actually want to look at the basically the distance between segments of these signals. Typically, you look at, for instance, absolute distance or Euclidean distance, and what we introduce is to look at the inner product between the signals taken as vectors, and this is less sensitive to variations in amplitude because these signals are show very high variations and artifacts in amplitude, also very strong spikes, and we wanted to see whether this type of approach was better. And in fact, it actually boils down to applying the Euclidean entropy to signals that are normalized in amplitude. So what we did is this trial. So we have this, what we consider as preterm birth is before the 37th week, and then the last 7 days we consider it labor. So these patients went to the hospital because of preterm contractions. We did the recordings with the same system, so this NEMO patch. So this is the same protocol that I've shown you. We concatenated signals coming from contractions and then estimate the entropy only on contractions. And you can imagine this is quite challenging part because this contraction, even by eye, are not that visible. There were in this case, two readers, and also with some support from tokodynamometry, etc., but still quite challenging to label these signals. This is the distribution of the signals. So you see, here is the threshold between preterm and term delivery, so after 37 week and before 37 week. And here is you see the measurement, the time of the measurement to delivery interval. So these were measured within 1 week to delivery, for instance. So these were clearly all preterm deliveries. And the measurement was already done in labor, so they really delivered within 1 week. And here you see, in fact, that if you want to measure labor, then in fact using the inner product as a distance doesn't work that well. So probably therefore labor is we can conclude that it's more related to amplitude changes in the signal. Instead, it works very well when we want to predict preterm delivery. That means that the pathway leading to preterm delivery has a special definition, as really a special pathway that we can recognize by looking at entropy discarding amplitude information. So we did so this is the statistics with a k-fold cross-validation. So this is by the cross-sample entropy and the multi-entropy, so multi-channel entropy that we see compared with the correlation coefficient that was already proposed, and the single-channel sample entropy that was the best indicator for labor that we had found. And you see that by multi-channel sample entropy, you could in fact statistically produce better result than by the previous indicators. This actually did not happen in the case of prediction of labor, where the most promising indicator is still a single channel based on approximate entropy. And the multi-channel also performed well, but we didn't manage to actually improve what we had already achieved with one channel, which is actually good, because then one channel may be enough to predict preterm birth. So it actually simplifies our application. Nowadays, we are also developing this technique for, in the case of in vitro, in particular in the case of in vitro fertilization, because infertility problem is now touching about 20% of couples that want to have a baby. So it's an increasing problem because of the trend to postpone childbirth. And as a result of that, many people have to, as a last resort, have to go through the in vitro fertilization procedure, where the embryo is prepared in vitro and then transferred into the uterus. And what happens is that uterine contraction most probably play an important role at this point, because depending on the amplitude and the direction of this contraction, you can have a successful or not successful fertilization. And please consider that only, despite the cost of about 5,000 euros per procedure, only 30% of the procedures succeed. So it's a very low rate, and everybody's trying a bit to understand what is the reason for these failures that are still not explained. And what we are doing is actually to analyze the activity of the uterus in this context, because this, as I told you, this contraction could push the embryo outside. That will, especially electromyography, I think it's a good example of that. It will produce a lot of additional data. These are even multimodal data, because we are trying also to acquire simultaneously signals from, for instance, accelerometry, in order to deal with artifacts. I haven't got time to discuss that, but that's also very valuable. So I think that next to this physics-driven modeling, that is the part that I've discussed now, I think it's quite important to develop also models and approaches for accurate, in real-time characterization and analysis and classification of this data, so that they can really be used for diagnosis, but also, for instance, for alarm systems, like in pregnancy. That would be very relevant. So thanks.
Thank you, Massimo.
For your attention.